A vaccine is a pharmaceutical composition that aims to awaken an immune response in an individual to whom it is administered and in this way protect it from infectious diseases. Between the 18th and 19th century, vaccination mechanisms were understood thanks to the efforts in which several scientists contributed knowledge on the way in which biological samples of diseases, which could be considered attenuated by their origin, served as preliminary vaccines for various diseases. Such was the case of the efforts made by the French army in the time of Pasteur and by the American armies during the Civil War. A vaccine is only as effective as its application in large population samples, since the basic principle on which this is based requires that to stop the spread of an epidemic, a vaccine is applied to the entire population. During the last 100 years, numerous infectious diseases have been eradicated thanks to global vaccination efforts. Such was the case with polio and smallpox. Furthermore, in the last 2 decades, there have been advances in the field of biotechnology that have allowed the development of new procedures through the use of genetic engineering and bioprocesses for the production of genetically modified vaccines. There are two large groups within the vaccine production process: the methods that involve the cultivation of the infectious biological agent, either in cell cultures or in embryonic egg platforms, as a method of bioproduction and propagation of the infectious agent. On the other hand, there are methods that involve the use of genetically modified crops for the production of key components of the infectious agent, which can be used as an active ingredient in the composition of a vaccine. These last procedures were derived with the boom of biotechnology at the end of the 20th century and therefore are still in early stages of development.